Good morning, everyone. Good morning, my advisory. We are still stuck in monohybrid and dihybrid process. Again, these are the bases or the basics or fundamentals of genetics. We'll be having a tool, okay, in determining the possible offspring. The tool that we are actually using is a Punnett square, okay? Again, when you say monohybrid cross, it is a cross between two organisms with different variations. Different variations meaning to say that they are not the same. Okay, they are different variants. Okay, variations, variants, those are just terminologies again. Have you heard about variants? The word variants.
Yes,
Okay,
yes,
variants means kind. Okay, variations. Okay, different strain. Like for example, in the COVID, okay, we have a new variant which is called the Omicron. Okay, Delta is another variant. So these are different variations, right? Of at one genetic chromosome of interest. Like for example, we'll be focusing on the pods, the green and the yellow. Let's have this human. Okay, these are plants, correct? But let's have this as example as a human, right? Human genes. So let me share my whiteboard. Okay. For example, we have um can you see my shared screen now? My whiteboard?
yes,
Okay. For example, we will be checking on the um the color of the eyes. Okay, the blue and the yellow, the blue and the black. Okay, let's take a look at um skin na lang skin. The white and the brown skin. Okay. White, this is the white, okay, because white is dominant over the brown skin. Okay, small letter W, it's brown. Hang on. Right? Okay, again. Um, Again, small letter W is brown skin. Okay. How about we have a cross between a heterozygous white, okay, heterozygous white, so meaning to say that the variant here or the variation here is carrying a dominant white and a recessive brown with a brown female, okay, with a brown female. So let's have this one. Let's use a Punnett square. Brown female simply because it carries only brown. Okay, it carries only a brown gene. All right. So if you're going to look at this, the male we have here male and female. Okay, the male is carrying a dominant white allele with a recessive brown skin allele. Okay, while the female here is a what? Totally a brown what? Totally carries a brown gene. There is no such thing as homozygous here homozygous brown simply because this is what? Recessive gene. Okay. Again, the recessive gene can only manifest, we can only just see a recessive gene if it is paired again with another recessive gene. Okay. So let's take a look at this one. To be, we call this one, to be uniform. Okay. In writing, in doing the Punnett square. Okay. Always and always, the area here should be female and here should be male. Okay, just to be uniform. Okay, for our male we have W, so we have a heterozygous white. Okay, this is common. Why? Sure. Where can we see this one? Okay, usually foreigners like those variation with a parent with a Filipino, we have these are in the case of half, okay, half German, half American, so they actually carries a recessive allele from their parents because they're not pure, okay, they're not pure Americans, pure German, okay. So in this case, if you are mixed with another blood, so you are carrying a recessive gene, right? So for female, we have all small letter W. Okay. So if you're going to match, okay. By the way, the one that they are using is a Punnett square. I have a lesson before. Okay. This is included in the lesson before when you were still in grade 8. Okay. This is a continuation of the lesson. Right? Okay, let's continue. Capital letter W. Okay, with a small letter W. This is heterozygous white. Small letter W and small letter W. So this is brown. Okay. Capital letter W. Okay. And small letter W. This is heterozygous white. Okay. And all small. Okay. Letter W. So this is brown. So what is the result now? In the exam, I'll be asking you about the phenotypes and genotypes, or phenotypic ratios and genotypic ratios. So let's start with the genotypic ratio. Genotypic ratio, okay? What is the genotypic ratio here? Genes. If you're going to talk about genes, okay, you are talking about if it carries a dominant allele or it carries a recessive allele. So heterozygous and homozygous comes in. Okay, this is where heterozygous and homozygous comes in. When you say heterozygous, means 2 alleles or more. That's heterozygous. When you say homo means 1. Okay, homozygous means it carries only 1 gene. Okay, like for instance, but homozygous is not actually applicable if you're dealing with a recessive allele or recessive gene, right? For this cross, okay, we can have here 2, okay, all big letter W and small letter W. This is heterozygous. This is 2 heterozygous white, okay, with a white skin. Heterozygous white is 2 because this is in ratio form. Okay, if it is in ratio, then you can actually do this one 2, 1, 2. Okay, 2 brown skin. Okay. Again, the genotypic ratio is 2:2. 2 heterozygous white and 2 brown. Or you can do this one. Okay. You can do the numerical form and you can also do the percentile or percentage form. So like for example, this one, 50% heterozygous white is to 50% brown. Okay, 50% heterozygous white is to 50% brown. So all in all, if you're going to have a total of it, it is 100%. Okay, how about the phenotypes or phenotypic ratio? Let's have the phenotypes. And when you say phenotypes, these are the observable traits. It doesn't matter if you're actually carrying a recessive allele, as long as it manifests into you. So those are just the one you can actually observe. Traits, behaviors, what else? Physical appearance, color of the hair, color of the skin, eye color. So those are the phenotypes. Okay, let's have the phenotypic ratio. Okay, phenotypic ratio. For the phenotypic ratio, these are the one you can actually see. Okay. In this case, I can actually see 2 white. Okay, is 2. This one is brown. 2 brown. Correct? Because we are actually thinking about what we can observe or the observable behavior, the observable traits. Okay. Or we have 50% white is to 50% brown. Right? Question so far? Do you need another
None.
example? Did you get the point? Everybody? Did you get the point?
Yes.
Okay, let's have
Yes.
another example. How about this one? The color of the hair. Okay, the black and the brown. Color of the hair. Okay, color of the hair is dominant black, capital letter P. Okay, with a recessive brown, small letter B. Okay, how about if we have this one, 2 heterozygous? So the male here is heterozygous black, and the female here also is heterozygous black also. What could be the possible result? Okay, this is actually in the 3rd generation. Okay, the 1st generation are pure. Okay, 1st generation are pure. The 2nd generation or F2 is heterozygous and mixed with a recessive allele. Okay, the one that we are actually we have a while ago is a cross between an F2, okay, or the 2nd generation. Let's have the F3 or the 3rd generation. Okay, for the F3, if you're going to look at this, what could be our possible offspring? So let's use the Punnett square. Okay, again, male, we have heterozygous black because it carries a gene for black, a gene for brown, and a heterozygous another black because it carries again a gene or allele for brown. Okay, let's have this one. We have capital letter B. Okay, you look at the box, you look at the motion of my cursor. Okay, BB. Okay. Always again, if in writing, okay, in writing these genotypes, okay, always start with the dominant allele. There's no such thing as B. Why? Because dominant allele should start first. Okay? Remember, it's dominant. The word dominant. Okay? Dominante. Okay? Dominante in Spanish. Dominante meaning it's what? Dominant. It exceeds the another one. Dominant. Okay? It exceeds the other one. Okay? So, this one is heterozygous black, another heterozygous black. Okay. Look at capital letter B and small letter b. And this one is a brown. Okay. If you're going to look at it, it is what? It has a lot of variations. That is why F3 is really good if you're going to ask me about the dispersion of the genetic characteristics. Best genes comes from the F3. Okay. Again, best genes comes from the F3 or the third generation. Right? Okay. So let's move forward. Let's have the genotypic ratio first. What could be our genotypic ratio here? Anyone or anybody here who can give me an answer? What could be our genotypic ratio here? Right. So for the genotypic ratio, we can actually see here. Okay, this one is homozygous. Homo means one, right? Correct? This is one
Yes.
homozygous because it carries only one gene, correct? Or one gene, rather. Homozygous black, okay, is to 1, 2, okay? 2 heterozygous black is to, how about this one? How about the last one, this one? It is 2 or 1 rather, 1 brown. If you're going to observe it, okay, brown can only be manifested, okay, it can only manifest if paired with another recessive allele. Okay, the absence of one, okay, the absence of one can mean that you cannot observe this behavior. Okay, it should be paired with another recessive allele. So in the progeny, if you're going to look at the progeny, you can actually see what? 1 homozygous black is to 2 heterozygous black is to 1 brown. So this is 1 is to 2 is to 1. So if you're going to look at this, this is 1 is to 2 is to 1. 1 homozygous black, 2 heterozygous black, and 1 brown. Okay, or we can have this one. Okay, you can always use the percentage if you want. Okay, 25%. Okay, homozygous black is to 50%. Okay, heterozygous black is to 25% brown. 1:2:1. That is the genotypic ratio of the cross between two, okay, heterozygous organism or two heterozygous alleles. How about the phenotypes or phenotypic ratio? Pheno means the physical, the one you can actually observe, regardless with their genome or genes. Okay, phenotypic ratio. Okay, 1, 2, 3. Of course, you can actually observe this one as black. Heterozygous black is also black, and heterozygous black is black. Okay, 3. Okay, 3 black. You will not identify them as homozygous or heterozygous as long as the appearance is black. Okay, 3 black, of course. This one, the 3 of them are all black. Okay, is 2. 1. This is not considered as black, correct? 1 brown. Okay, or you can have this one as 75%. 75% black is to 25% because this one brown. Okay. Again, the phenotypic ratios are 3:1. This is 3:1. Again, when you say phenotypic ratio, do not be confused with the terminologies. Okay. Again, associate things over. Okay. When you say genome or genotype, okay, you always get the root word genome. Genome means genes. So these are the genetic characteristics. These are the genes. So you need to identify if it is a homozygous or a heterozygous. That's in the genome. Okay. How about in the phenotype or phenotypic pheno? Okay. Phenotypic ratio. When you say phenotypic ratio, it is the appearance, the one that you actually observe, the observable behavior or traits. Okay. So in the genotypic ratio, we have 1:2:1 or 25% is to 50% is to 25%. How about in the phenotypic ratio? We have 3:1 or 75% is to 25%. Did you get now the concept behind? Everybody?
Yes.
Okay. These are the bases, right? Please do not forget this concept. Why? Because we'll be using this one all throughout. Okay. Let's tackle now. Anyway, you can always visit this video because I'll be uploading this one in your LMS. Right? Let's tackle now the dihybrid cross. Okay. Di means 2 or more. Di means 2. Okay. Dihybrid cross. We already done this one, but we usually use what? The genes for humans. This is in the plants. Okay. How about dihybrid cross? Okay, dihybrid cross is a cross between two different lines or genes that differ in two, okay, observable traits. Let's use again another human, okay, human traits, so that you can always associate things over with this, right? Okay, let me share my whiteboard again. My whiteboard is back again. Okay, let's have a cross between, again, 2 or more genetic interests. Okay, 2 or more. So meaning to say we can have the color of the hair. Okay, let's take a look at this one. Okay, color of the skin and color of the eyes. Let's have this one. Color of the skin is white. Dominant white. over recessive brown. And the second allele is the color of the eyes. Okay, color of the eyes, usually blue eyes is dominant over brown. Or let's have blue eyes with let's not let's just use black here. Okay, blue and right, blue and shall we say gray. For example, we have the two alleles here with two genetic interests. Okay, a cross between, like for example, a heterozygous, a homozygous white. Okay, homozygous white with homozygous blue eyes to a, again, let's start with the skin, homozygous white, okay, with gray eyes. Sure, how can we get the possible, okay, the possible variations in here? Please take note that in crossing dihybrid, everybody, are you still with me? Everyone?
Yes.
All right, in crossing dihybrid, you need to cross this first, okay? A cross tagsa-tagsa. So how can we do this? Okay, you need to have a separate Punnett square. So cross WW, capital letter B. Okay, so we have, let's start with, again, an allele, which is the from the skin. Okay, we have WB is to WB. All right. In this case, you can actually use the current setting that you are actually having. So we have BW, okay, BW, BW, BW. Why? Let's take a look at later, okay? Because this is not just, this is not actually part of the dihybrid, the dihybrid offsprings, okay? This is just variables, right? So we actually get this from male. So the crosses or the variables that we have, okay, 1, 2, 3, 4. We need to actually put it separately. Hang on. 1, 2, 3, and 4. So let's have this one. These are the variables. So the variables will be added here. BW, 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 and BW. Correct? Are you still with me?
Yes.
All right. So we are done with the first, or the male. Let's move now to the female. Okay. Same, again, same Punnett square. You need to cross it over first to itself so that you can actually see the variables. These are the variables, right? So we have a homozygous white with gray eyes. Okay. We have WB, 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 W. Okay. Or BW, small letter BW, or small letter BW. So let's have this one, WB, WB. Okay. WB, WB. there you go. We did actually extracted, okay, again, we did actually extracted the variables of the crosses or variables of this organism or the two alleles, okay? What's next? If you're going to observe, we started with white, correct? We started with the color of the skin and the color of the eyes. Okay, so let's have the color of the skin. 1 and 2. This is homozygous white, okay, with heterozygous brown, okay. We have again color of the skin. We have heterozygous white, homozygous white with heterozygous blue eyes, okay. How about this one? Again, we have heterozygous white with wait. Again here, if you're going to look at this one, we have capital letter W and small letter W